A very good evening to everybody, and welcome to another episode of The Different News. Today, I have Ramesh Jayaraman, the founding director of DOSE Quantics Consulting. Welcome, Ramesh, and thank you so much for being on my podcast and accepting my invitation. It is a pleasure having you here and knowing your journey.
Thank you, Dr. Gargi, for the invitation. It's a pleasure to be in this program as a guest of honor.
Thank you. So, Ramesh, before we begin, and you are having such an extensive academic and professional journey, please tell us about your journey and how you landed up with DOSE Quantics Consulting.
I began my career as a biochemist 30 years ago. I have a master's degree in biochemistry from the University of Agricultural Sciences in Bangalore. At that time, I wanted to do a PhD further in biochemistry, but as circumstances would have it, I entered the industry instead. And that was my first step into the pharmaceutical industry. I joined AstraZeneca. At that time, it was Astra Research Center India in Bangalore. And Astra was a non-profit organization doing fundamental research in malaria and tuberculosis. This is 1994. Then eventually it became AstraZeneca. So I joined as a biochemist and worked on biochemical targets in drug discovery. That was my first step into the pharmaceutical industry. As you know, drug discovery is a very long process. It takes probably 10 to 15 years to bring a molecule to the market with just around 10% success rate. It's a long journey, it's expensive, it's risky as well. So when I joined Astra, I began my career as a biochemist, purifying enzymes and characterizing. These enzymes are what we call as specific targets, say in bacteria. Where I was working, it was in tuberculosis. So we had to identify these enzymes and design molecules which inhibit those enzymes. And then we hope to inhibit the bacteria, kill the bacteria, and then take it forward from there. So I spent a few years in isolating these enzymes from the bacteria called Mycobacterium tuberculosis, which causes TB in humans, as well as from bacteria called Mycobacterium smegmatis and E. coli, which is a common resident. I also entered into cloning and expression of these enzymes, which we call as molecular biology nowadays, recombinant DNA. And after 3 to 4 years, there was a necessity for doing pharmacokinetics. That is, you take promising molecules and see whether they are drug-like. Whether, suppose we dose an animal, whether you see concentrations in the blood that would give efficacy, say in a TB model of, a mice model of TB. That is the next step. So I began to start doing pharmacokinetic experiments. I learned it. During my job, I learned that was not my field of expertise. So it was like learning a new subject. It was very exciting for me. At the end of the day, pharmacokinetics is intimately connected with biochemistry. In living organisms, all the molecules undergo synthesis and degradation, including whatever food we consume. There are different molecules, say vitamins, lipids, carbohydrates, whatever we consume, they are all absorbed into the body and then eliminated. And they also get integrated into the body. Similarly, for foreign molecules like drug-like compounds, you have to ingest them and see how they perform. Only if they are drug-like can they move to the next stage. So I began training in PK and worked as a PK scientist, eventually as a pkpd scientist in AstraZeneca for 14 years. My dream was to see whether we could get a drug out of AstraZeneca India for the treatment of tuberculosis. But unfortunately, we could not move molecules beyond a certain stage because TB already is a very tough disease. However, having said that, I was involved in characterizing the pkpd of the frontline TB drugs like rifampicin, isoniazid, ethambutol, and pyrazinamide. And they were not well characterized at that time. So we helped characterize it and form the basis for evaluating all other compounds in the TB project. But it didn't work out to be a drug in the market. So I was a little dejected at that time. After 14 years, it was a tough thing. I thought I would retire from AstraZeneca, seeing many compounds become drugs. But unfortunately, that wasn't realized. Then I moved to SBio Singapore, which was a small biotech company, which worked in cancer discovery and development, small molecules, targeted therapies in cancer. That was a move where I went from a multinational company, a large multinational pharmaceutical company, to a small biotech company, around maybe 50 to 60 people at that time when I joined. And in this company, I worked as a senior DMPK or a PKPD scientist. But this time, we had 5 programs running. 4 of them were at different stages of clinical development. And I was part of the product development teams for this. There were molecules like SB939, which was developed for solid tumor cancers. There was SB1518, which was called Pacritinib, and which went on to become a drug called VONJO. I will talk about that in a short while. Then there was 1578 for rheumatoid arthritis. And then there was SB1317, which was for multiple cancers. So what I'm trying to say is I was able to gain experience in clinical as well as preclinical development of these molecules. As you know, drug discovery is a multidisciplinary approach. A single person cannot do all the things. So we need different disciplines. And one such discipline is pharmacokinetics and dynamics. And amongst the 5 programs, SB1518 was developed in SBio. It was discovered. It came from a novel class of molecules called macrocycles, which we developed in the non-clinical models. And then we moved to phase 1 and phase 2. Then eventually it went on to become a drug. So the dream which I was talking to you about, which I could not realize from AstraZeneca, was realized in 2022 when we heard that it was approved by the FDA for the treatment of myelofibrosis, a rare blood cancer. So it was like fulfillment of a dream which I had. I was very happy, pleased, and it was also intellectually stimulating to see something which we discovered from a small biotech company has been approved for treatment of patients. From the community point of view, we had the sense of satisfaction that we did something which benefited patients. At that time, myelofibrosis, for which Pacritinib or VONJO was approved, was an orphan disease. There was no drug to treat that disease. So in that sense, we were all very pleased to see that it was approved after 16 long years and after having spent more than $1 billion. And like I said before, 10% success. And in cancer, it is even less. So despite all that, we were able to see it go to the market. So that was what you could say a highlight for my career. And the other compounds also moved to the clinic. Some made it, some did not make it. But The environment in a biotech pharma is far different from a big pharmaceutical company. You learn to interact with multiple departments, interact with people, you learn to make decisions in drug discovery and development. That was my SBio experience. I came back, worked briefly in GE Healthcare for developing these tracers which are used in PET, CT, MRI scans. After a couple of years there, I was consulting for different companies before I started TerraIndex along with my colleagues in Bangalore. TerraIndex was founded in 2014, which is a company which provides services to the pharmaceutical and biotechnology companies, testing at various stages, which I had mentioned to you before, in the non-clinical side, not on the clinical side. From TerraIndex, I had the opportunity to work as a chief scientific officer and collaborated with many clients. And one client, uh, we had a good program where we established an animal model for infection in rats, and that was used to progress their molecule. And today that molecule is in phase 1 trials. That's another example of being associated with a molecule that is going into the clinic. We had another client for which we did work, and their molecule also is in phase 1 trials. So for a pharmaceutical scientist, this gives a tremendous sense of satisfaction when you see molecules becoming drugs, or at least on the way to becoming drugs. I spent 10 years after having co-founded TerraIndex, and now recently I started Dose Quantics Consulting, which is a very new company which provides consultancy services across drug discovery, especially in PK and pkpd. That's my journey so far.
It's an amazing and very commendable journey, and the kind of experience you have gained through all these years. I think it's something very inspiring for people who come from the pharmacology background also, or biochemistry, and the way you took step by step ahead, and you have such a flourishing career, and today you have found those Quantics. I think this adds value to the young aspirants. They look up to you, people like you, and I'm sure that they get inspiration from your journey, of obviously. Ramesh, you did you find any particular area of gap when it came to drug development and discovery consulting, which probably inspired you to bring forward those Quantics? Is there any specific gap that you noticed?
Yes, like I said, drug discovery is a very long process and it's a risky process, and critical decisions have to be taken as you move molecules from discovery to into development. In India, R&D in pharmaceutical industry is not very big. There is no environment to be patient to do drug discovery. As I said, it takes time. You can fail at any stage, and there is an unwillingness to get into R&D. It's largely now driven by generic drugs. So nevertheless, there are companies doing R&D in India, many startups, and there are pharmaceutical companies which are doing R&D. And there are many companies amongst the startups which develop very nice compounds, discover new compounds. It could be molecules, cell therapies, biologics. But there seems to be a gap. That's what I perceive in taking it to the next stage, like doing pharmacokinetics, how pharmacokinetics impacts efficacy, the pharmacodynamics, I mean. So many compounds have to be evaluated based on pharmacokinetics and dynamics. In other words, to put it simply, are they drug-like? All compounds or biologics are invented or synthesized in the laboratory by testing on specific cells. It can be microbial cells, it can be human cells, animal cells. It may show good activity there, but that is necessarily not the final thing. It has to become a drug. And for that to become a drug, it takes a long time, and it has to enter the body, reach the site of the disease. It can be infection, cancer, it can be Alzheimer's, then act. So not all molecules will make it. And that's where PKPD comes in. Scientists like me step in and tell what is needed to move to the next step. I saw this gap and I thought that I should fill this gap. That's the basis for starting those Quantics.
Amazing vision. So, Ramesh, can you just brief us on what kind of services those Quantics Consulting provides in the field of PKPD and pharmacokinetics and pharmacodynamics?
There are many services. The first service is that those Quantics can engage with clients from the beginning of the drug discovery programs, especially in decision making based on all the data that is accumulating, and more specifically PKPD data. In PKPD, there are three verticals: PK, PD, and PKPD. So PK is pharmacokinetics, as I told you. PD is pharmacodynamics, that means the efficacy, what the drug does to the body. PK is what the body does to the drug. So put it simply. And PKPD is linking the concentration to effect. The effect can be efficacy or it can be toxicity. So in these three, those Quantics helps clients analyze PK data using specific softwares. Not only analyze, but also give interpretation of what that data means to progress the molecules. The second is pharmacodynamics. We assess the capability of a molecule to demonstrate efficacy in the lab or in the animal models, or the potential to show activity in humans. So a compound has to be tested in both these systems. Eventually, both have to be integrated. That's what we call as PKPD. And PKPD helps in setting dose to be given, the dosing frequency, number of times the dose has to be administered, and the duration. All these three matter in setting dose in humans. So that's vital. So those Quantics provides consultancy in these three verticals, which in turn link to the drug discovery process.
Thank you, Ramesh, for giving that extensive but very simple overview of what those Quantics is providing to people, and I think it's fantastic. So I'm sure, like being in such an extensive career in your entire journey, can you tell us like some challenges? I mean, most prominent challenges that you, as a scientist or as an entrepreneur, you have noticed in this particular field of drug discovery and development?
So there are three main things which I have observed in drug discovery while working in a big pharmaceutical company and working in a small biotech company. To do drug discovery, it's important to have capital. There has to be investment. Otherwise, it will not work because it is expensive and it's risky. So the companies must realize that you will invest, but you may not necessarily get a drug out of it. That is one thing. You must be able to invest. The second thing is that you have to bring in people from different disciplines and work as a cohesive team to move the molecule across different stages. This is a very challenging aspect of drug discovery, making everybody see the bigger picture, where we need to go, rather than getting stuck on small issues. You may not have 100% of the data to make a decision sometimes, depending on where you're working. If you go on fine-tuning one specific parameter, you will be delayed in optimizing other parameters for a molecule. So delays will happen, and it'll unnecessarily delay the time to the clinic to meet a clinical need. The third is a clear vision of where you want to work. You can't lose focus. If you have identified an area where you have to work, you have to be focused on it, and you should not lose the focus and say halfway down, I'm not interested. It does not work that way. Or if you should remember that drug discovery development is a race, because many companies may be working in the same area as you may be working. So you must be aware of what is going on, constantly monitoring what is happening out there. So all these forces are always acting on the drug discovery team. So they must be able to adapt and be flexible to these. And many times this may not happen, which can cause closure of the project. So you have to be very clear about all these things. If these things are not met, and if critical decisions are not taken, program may fail.
Yeah, I think it's the practical things that you have seen, and I think these insights are really helpful, because I think these are the points where even companies who are existing, or probably who are trying to, get into the field with much enthusiasm, these are really helpful insights for them. Absolutely. So, as we see this, the drug development, discovery, and the field of pharmacokinetics and dynamics as such, so is there any particular technology or any particular area that you see coming in the near future in this field, and that excites you? I mean, can you enlighten us on this aspect as well?
Yes. Now, PKPD is becoming increasingly important for regulatory agencies like the US FDA and EME, European Medicines Agency. They request or they ask for PKPD data during submission of, say, an IND package, that is, investigational new drug. The reason they ask is that we should be clear about the relationships between PK and PD, which is the basis for setting the dose regimen, and duration, as I had indicated earlier. The second thing is that they also increasingly look for modeling and simulation of PKPD. That means you employ mathematical models to predict pharmacokinetics and pharmacodynamics in humans before the clinical trials actually begin. The modeling and simulation help in understanding whether the candidate drug which the company is going to evaluate in the trials, will it show the chance of reaching the target exposures for efficacy? Would it show any adverse events in terms of drug-drug interactions? All these data are very valuable for them to arrive at a decision to authorize clinical trials. And it's becoming increasingly evident that PKPD modeling and simulation are playing a very critical role. There have been a lot of examples where the FDA have used modeling and simulation of PKPD to avoid expensive clinical trials. The PKPD modeling data are shown in the prescription information of the drug, saying that based on so and so PKPD approaches, this is either contraindicated or can be administered in specific populations of patients. And expensive clinical trials have been waived off based on this. So that's the power. And the field is evolving rapidly in fields such as physiologically based pharmacokinetic modeling, which we call as PBPK. Then there is quantitative systems pharmacology, which integrates all the host factors, the population factors, and drug factors, and predicts whether a molecule is capable of showing efficacy or not. And this becomes increasingly important in precision medicine. That means one drug does not fit all. A drug fits a specific patient, and the dosage will be tailored to that specific patient. So precision medicine is advancing rapidly, and PKPD is one of the technologies that is helping to accelerate it. And of course, artificial intelligence has come in, and that is integrated with the PKPD modeling and QSP modeling, what we call as pharmacometrics. And this, when integrated, will speeden up the process, I believe, to set the dose, rather to set the optimum dose for the patient or a patient. Like I said, a single dose may not be suitable to all the patients. There could be adverse events due to their disease conditions or their demographics. So it's important to understand the patient characteristics before you arrive at the dose. So this field is rapidly advancing. You will see this being increasingly used in packages submitted to regulatory agencies and how they evaluate. So it's exciting times for PKPD.
Excellent. I mean, very many new information I'm learning from this conversation, and I'm sure our audience is also learning a lot. So, just taking a trail of your previous, answer or the insights that you shared regarding the regulatory part. So, does those Quantics help? Is there any workflow that the clients who come and work with those Quantics you help in, in this path of regulatory process to them as well?
Yes, those Quantics engages with clients to see if they have the relevant PKPD data that is required for regulatory submissions from non-clinical studies. Those Quantics helps in design of those experiments, interpretation of the data, and how they will be used, how they should be used during the submission of the process. That is one part. The second part is prediction of the first time in human dose. This is the transition where a clinical drug candidate goes from non-clinical stage and is dosed in humans for the first time. When it is dosed in humans for the first time, it is important to identify the optimum dose to begin with, because we don't know what may happen in humans. Even though you have a lot of non-clinical data, you need to know what is the optimum dose. And PKPD helps in setting that first time in human dose. So Dose Quantics engages with clients to help predict those doses. It's also possible to predict the concentration time profile in humans based on modeling. This is another service that Dose Quantics provides to clients. All these are done, you must remember, with the main goal to reduce the chance of failure, or rather increase the chance of success of compound going from non-clinical to clinical, because most of the failures happen at this stage.
True, very true. Yeah, I think that's amazing. And I think those Quantics with your services, you are catering to all the problems that people can face in these drug development area. I mean, taking it forward, particularly the translational part when it comes from non clinical to clinical. So I think it's amazing. So I think I think in this journey, like as you mentioned before, it's we need partnerships and collaboration. So what would be your take on the importance of collaborations in bringing together, data or people in actually solving the challenges and really fast tracking the process of taking a molecule to more into the success points. So, what would be your take on this?
Yes, like I had mentioned earlier, drug discovery development is a multidisciplinary approach involving chemists, bioinformaticians, biologists, pkpd scientists, toxicologists, clinical pharmacokinetics, pharmacologists, rather, QA, QC. There are many departments which have to work together. Now, in big pharmaceutical companies, all these departments are there which have to work. But say for smaller companies, all these departments are not there. It's not possible to have all of them. So they have to collaborate with different disciplines. And in this model, those Quantics collaborates with clients or engages with clients for PKPD purposes as well as drug discovery and development from the starting to the end. And it can be in different ways. Either it can be just a service or engaging with the client from the beginning so that you know the program, you're with the program, and you go along with that from discovery all the way till development. That is the way those Quantics are, and I would like to work. But oftentimes it may not happen. You get involved in specific collaborations for specific projects. But collaboration is the cornerstone for drug discovery and development. For any therapeutic modality, it is very important. And you can see it happening. Companies will collaborate with other companies which have their expertise.
Right. So, just a little bit, another question related to collaboration. So, as you said that, big pharmaceuticals have their, I mean, there's a setup there. So, I mean, is there any, from your experience, do you have any suggestions or advice like how small biotech companies could approach these big pharma companies, or probably equal businesses, so that they survive as well as they can be backed up with the power the other big companies have?
Okay,
Any
so
suggestion? Yeah.
in this context, what happens is, or at least where maximum collaborations happen, is when a small company has a very novel product. It can be a molecule, it can be diagnostic, it can be small molecule or diagnostic, which is novel, and they have patented it, but they don't have the funds to take it to the next step. So, pharmaceutical companies nowadays are increasingly looking to smaller companies which have specialized products but which cannot take it forward. So, they either acquire them, or they pay them royalty and then acquire them, so that the company takes over the molecule and takes it forward. The small company gains revenues from that with milestone payments. So it's a win-win for both. The pharmaceutical company gets a novel product to take to the market. The small company gets funds or is paid for its work. Maybe it can start a new program. Many companies, small biotech companies, follow this model. And you can see in the press that some company or the other is acquired for not millions but billions of dollars for a specific technology. The latest example is antibody drug conjugates, which is a very hot field in cancer. So, a lot of big pharmaceutical companies are investing in ADCs, and small companies have developed technology to produce ADCs. So, that is one of the major collaboration models which are in vogue these days.
Thank you, Ramesh. That was very valuable insights. And well, we are almost at the concluding part of our conversation. Though I don't want to let you go because it's very interesting, and I'm learning a lot from whatever you are sharing. Um, so, what are your long-term goals with those Quantics, and how do you plan to expand or diversify your services in this area?
So my long-term goal for the company is to be a preferred consulting company for drug discovery development, specifically in the areas of PK and PKPD. That's my long-term goal, to be able to be there. The plans to diversify is to get more technologies into the company so that it can perform more advanced modeling and simulation and use tools to predict PK, PKPD efficacy and toxicity in humans. That would be the model for diversification. I also dream of starting R&D to develop new molecules, but that's a very long way off. You need to have ideas to start drug discovery on your own. But if somebody is there, I'll be very happy to be associated with them in taking the molecule forward.
Thank you, Ramesh. It was wonderful talking to you and knowing your journey and whatever scientific contribution and help you are bringing through your consulting company to the drug development and this portfolio. I think that's very helpful. And thank you so much for being here. I hope we'll meet in near future for further interesting conversations like this.
Thank you, Dr. Gargi, for giving me this opportunity to be part of your program. It was very nice to discuss different aspects of drug discovery about PK and PKPD, because this is not readily understood by many people. You need to explain this to convey a message what pharmaceutical scientists actually do. Thank you. I
Very
hope it is helpful for people to understand what we do.
Absolutely. That's you have explained in a very simple way. Like any layman listening to this probably would understand what you're trying to say. Thank you so much, Ramesh. Thank you so
Thank you
much.
so much.